Okay. Good evening. Good evening. Can you hear me? Oh, sorry, there is problem. Give me a second. Now, okay, right. Okay. Okay. Sorry, there was a problem in our Zoom account. That's why today late. Okay. Now, okay. Everything. Yes. What we discussed up to disaccharide, right? We discussed up to disaccharide. Okay. First of all, now 2025 exam, 2025 A-level exam, 2025 A-level exam was postponed to January, first week of January, the second part of the exam. Therefore, 2026 A-level, according to schedule, it should be postponed at least September now. Okay, September or first week of October. First week of October. Think it is September. If we think September, we have only 8 months because now this is December and another 8 months. Then overall we have 9 months now. 8.5 months. 8.5 months. Actually, this is advantage because in the previous we had only 7 months, 8 months. Now we have additional 1 month for prepare for the exam, September or October. Still not decided. Okay, it can be vary. Now don't have exact time. Now this time O-level exam also will be postponed due to this disaster. Okay, however, all works, all papers and all lessons we have to complete. At least we should have scheduled to complete all things at least before August. That's our plan. Before August, everything we need to complete everything. The second thing, this is actually last week class. Last week we missed one revision class. Okay, this is the last week class. This is the last week class. Again, you have no classes in Friday. Okay, Friday actually I'm too busy. Again, no classes Friday. This is the final class we're doing in Friday. This is your regular timetable. You have 8 hours per week for first few months up to April. up to April. Up to April, Tuesday. Now Tuesday don't have 2027 classes. All will be changed. It will be held in Sunday morning. I think you have all full timetable. Tuesday 6 to we starting 6 because today there was a problem. If not, we start at 6 p.m. 6 to 10 or 10:30. I can give you one interval in the mid of class. This is a Tuesday. Tuesday you have 4 hours, 4.5 hours. Then again you have Sunday. 2 to 4 p.m. Okay, this is your regular time from next week. Next week mean we start this Sunday. You have class. This Sunday you have 20 to 27 class එකක් තියෙනවා. Revision එකක් තියෙනවා. From after today actually. Today is the 5th, no? After today, this is the timetable, our regular timetable. Okay. After April, you have another 1 day for past paper discussions. Up to April, we're not going to discuss past papers. We discussed our papers, our model papers. From April, we discussing our papers plus past papers. Therefore, you have three days, 12 hours. Okay. Okay, what we discussed. Now, 2 weeks passed due to flood. Okay, we discussed up to now main characteristic of disaccharide. Am I correct? We discussed up to main characteristic of disaccharide. Yumidu? Dihini? Main characteristic of disaccharide, right? Yes, main characteristic of disaccharide. Okay. Before start, let's revise again what are the main characteristic of monosaccharide. Okay, this is monosaccharide. For refresh, okay. For refresh, this is monosaccharide. This is monosaccharide. This is monosaccharide. This is disaccharide. Disaccharide and monosaccharide. What are the main characteristic? Okay. If you got polymer or not, polymer or not. Sorry, first one. First one, polymer. Polymer or not. If you got monosaccharide, polymer or not. I need quick answers. Polymer or not. Monosaccharide, not a polymer. Monosaccharide mean, I think you can remember, Monosaccharide mean main building blocks of carbohydrate. That mean very simple structures. What are the meaning of polymers? Polymers mean combining lot of monomers, very complex, large structures we called as polymers. Then monosaccharide is not a polymer. Monosaccharide is not a polymer. But disaccharide, Fatima? Disaccharides? Disaccharides? Yeah, not disaccharide mean two monosaccharide combining with glycosidic bond, then produce a structure we called as propose structure we called as disaccharide. Last week we discussed all characteristic, right? We discussed what is the structure of lactose, what are the structures of uh, maltose, what are the structures of sucrose, lactose, uh, maltose, syllabiose, all disaccharide type we discussed one by one, and all structures we discussed already completed, right? Now don't forget, again and again you have to revise these things now, because you have limited time. The thing actually, our syllabus not too advanced, but what you need to do, you have to you should have a continuous practice with consistency until exam day. Continuously you should have to work. Okay, this is the requirement. Then the exam paper is just another type of paper. Okay, it's not a special one. Okay, the second one, if you got the taste, how about the taste of monosaccharide? Monosaccharide, glucose, galactose, fructose, fructose. How about, yes, sweet in taste? Good, sweet in taste. Disaccharide, yes, disaccharide also sweet in taste, because sugar. What's a disaccharide type of sugar? Normal we eating sugar is sucrose. Sucrose is sweet in. Sweet. Okay, the next characteristic, third one, uh, crystallability, crystallity, crystallity. Monosaccharide crystal or not? Monosaccharide crystal or not? Yeah, both are crystals. Very good. Both are crystals. Crystallable, yes. Crystallable mean they can make as crystals because they are very simple compounds, like sugar like crystal. Okay, both are crystal. Both are crystallable. Uh, Fourth one, hydrolysis ability. Hydrolysis ability. Monosaccharide, how about can or can't? Hydrolysis ability. Hydrolysis ability. Monosaccharide. Monosaccharide. Yes. Monosaccharide cannot be hydrolyzed because hydrolyze means break down again into simple compounds. Now the monosaccharide is a main building block. That is it already. That's already simple, right? Again, cannot be breakdown. Can't be breakdown. Disaccharide. Yes, disaccharide, two monosaccharide combined with glycosidic bond. Disaccharide can be hydrolyzed. Yeah. Disaccharide can be hydrolyzed. Okay, do you remember the final part of the last week lesson I explained? What are the methods we can use for this hydrolysis process? How we can use, how we can hydrolyze this disaccharide? What are the methods we can use? Yes, I told main two methods for hydrolysis. It is given in the initial parts of the tube in disaccharide part. Yes, we can use enzymes. Very good. We can use enzymes. I told final few minutes of last week class, I explained different types of enzyme for hydrolysis process of disaccharide. Okay, I told maltose we can hydrolyze by maltase enzyme. Yes. Lactose we can hydrolyze by lactase enzyme. Syllabios don't have special enzyme type. And sucrose we can, sucrose hydrolysis process we can use two main types of enzymes, sucrase enzyme and invertase enzyme. They are MCQ key points. Okay, always ask MCQ. Sucrose we can hydrolyze by sucrase enzyme and invertase enzyme. Sucrase and invertase enzyme. Sucrase and invertase enzyme. Okay, if we not going to use enzymes, we can use directly diluted acid, diluted powerful acid like hydrochloric, HCl, sulfuric, nitric type powerful acid diluted solution we can use for hydrolysis process that I have explained in the experiment in our next part. Okay, any disaccharide, if we add any powerful acid and if we heat it, then it is, or it's auto hydrolysis breakdown. It's breakdown by using this acid and if we heat it. Okay, hydrolysis ability. Final one. Final one. Reducing or non-reducing sugar. Sugar type. Sugar type. Sugar type. All monosaccharides. Very good. All monosaccharides are reducing sugars. Reducing sugars mean simple sugars. The meaning simple sugars. Reducing sugars. Reducing sugars. Reducing sugars. Disaccharides. Can anyone tell me disaccharides, about disaccharide? Yes, all disaccharides, all disaccharides are reducing sugars except sucrose. Remember, only sucrose non-reducing sugar. Other all disaccharides are reducing sugars. All are reducing except sucrose. Sucrose නැතුව අනිත් ඔක්කොම reducing sugars. Reducing sugar mean simple sugars. Non-reducing mean bit complex sugars. Okay, that's a mean. Okay. Okay, that's all these characteristics we discussed, right? Water solubility. I forgot. The final one. Water solubility. Water solubility. What do you think? Yeah, both are soluble. Both are soluble. Yeah, I think you can understand. If you got monosaccharide, glucose, fructose, galactose, they are easy. Glucose, fructose, galactose, they are easily soluble in water. If you got disaccharide like sucrose, lactose, they also soluble, because if it is not soluble, we can't make tea. No, how we can make tea if it is not soluble? If you got milk sample, lactose is soluble in water, right? The milk sample is a water mixture with water. It's a water soluble. It's water solution with mixed with lactose, different casein, protein, and everything mixed. Okay, then it is soluble. Okay, they all are soluble in water. All are soluble in water. Okay, they are the main characteristic we discussed between monosaccharide and disaccharide in the last week class. Okay, any doubt up to now? This disaccharide you can ask now. I can give 1 minute. Any doubt? Everything clear? Okay, let's move into third types of carbohydrate. Still we discussing carbohydrate. Okay, this is a final part. After that, we can move into our protein and lipids, enzymes and vitamin. That's the end of our lesson. Okay. Okay, polysaccharide. What is the meaning of this polysaccharide? Polysaccharide mean, I told the main building block is monosaccharide: glucose, galactose, fructose, ribose, ribulose, deoxyribose. I think you can remember. In monosaccharide, I told different types of different classification of monosaccharide according to the number of carbon, according to the functional group, aldose and ketose. Okay, everything you should have to remember. According to functional group, what are the glucose and galactose, aldose, fructose, ketose? I think you can remember. And according to the carbon type, triose, tetrose, pentose, hexose, heptose, they are the main types. And I given all examples for each type. Okay, then every types of monosaccharide, they are the main building blocks of carbohydrate. Main building block, that means initial, the like in our body, the cell is the main building block. Samely in carbohydrate, the main building block is the monosaccharide. Now what is the meaning of disaccharide? Disaccharide means two monosaccharides. This is a one monosaccharide. This is the main building block. Disaccharide means two monosaccharide combined with glycosidic bond. Glycosidic bond. This structure we called as, this one we called as disaccharide. Disaccharide. This is the disaccharide. Two monosaccharide combined with glycosidic bond. The formation of glycosidic bond I already explained last week. The same thing. The two monosaccharide combined with glycosidic bond, then the produce structure we called as disaccharide. Okay, the same thing. The polysaccharide mean monosaccharide polymerization. Monosaccharide polymerization mean more than actually large number of monosaccharide. Large number of monosaccharide. Make it in our syllabus, we not going to discuss more about oligosaccharide. Make oligosaccharide mean, see, monosaccharide mean only one. Mono mean one. Mono. Then disaccharide mean two monosaccharide combined with glycosidic bond. The make structure. There is another one, trisaccharide. Trisaccharide means they are not commonly available. 3 monosaccharide combined with glycosidic bond. There is another one, oligosaccharide. It is a popular one. Oligosaccharide means 4 monosaccharide combined with glycosidic bond. There are different types. Okay. Now the polysaccharide is another main type. This is polysaccharide. Polysaccharide means large number of monosaccharide combined as a chain or very complex structure by combining many number of polymerization of many monosaccharide. It is a large number of monosaccharide polymerization with glycosidic bond make very complex large structure we called as monosaccharide. The hydrolysis of polysaccharide. Hydrolysis means if any polysaccharide. Now polysaccharide made by polymerization of monosaccharide. That means large number of monosaccharide combined together. Then if we break down again, we can get building blocks. No, simple. We can get building blocks. Normally insoluble in water. This characteristic again we can discuss. All polysaccharide normally insoluble in water. Like if you got as example, cellulose is polysaccharide. Cellulose mainly, this is the main compound in cell wall, plant cell wall. Is it water soluble? No, if it is water soluble, with rain, plants are dissolved. Plants are soluble. Okay. They are not water soluble and they are not crystallized. They are not crystals. They are very large compounds. They are not crystals. And yeah, they are the examples. Cellulose, starch, glycogen are the example for polymers. Okay. Example for polysaccharide. Write down first one. Starch. Starch is a polymer. The second one, cellulose. Third one, glycogen. Fourth one, inulin. Okay, go to the tute. We are in page number. I hope you all complete the tute. Structures, don't miss any structures in previous parts. Okay, we are in the page number 11. Page number 11. I don't have I know inulin. 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 Okay, let's discuss about starch. These polysaccharide structures you don't need to know. You don't need to draw because they are very complex, large structures. You can't draw, but you should have very basic idea how they form, how this starch is formed. I will explain it. Simple. Okay. Now monosaccharide, sorry, polysaccharide. The first example is starch. Starch you already know, right? If you got what are the foods available starch, mainly available starch? What? Yes, rice, maize, potatoes. Yes, potatoes, potatoes, potatoes. Okay, first understand this basic. You already know what is the main product of photosynthesis. In photosynthesis, using carbon dioxide and water, they make C6H12O6 plus oxygen plus energy, right? This is the photosynthesis. The main product of this photosynthesis is glucose. Glucose is the main product. Glucose is a product in photosynthesis. But this produce glucose in photosynthesis process, what they doing in the metabolic activities, in the activities in the plant, it is converted into sucrose for their storage process. Sorry, not sucrose, starch for their storage process. Okay, therefore this starch we called as the starch is the main stored food in photosynthesis, plants and organism, photosynthesis organism. Okay, the starch is the main stored food in stored when may vidiyata main stored food in photosynthesis organism. Photosynthesis, why we call photosynthesis organism without call photosynthesis plant without saying only plants? Why we call the main stored food in photosynthesis organism? You know, some microorganism like cyanobacteria and algae, they can make foods, their own foods. They have chloroplast. Therefore, they can make foods. Okay, they are this microorganism like cyanobacteria, algae, they are stored food also. This is starch. Remember, the main function of this starch, they are the main stored food in photosynthesis organism. Photosynthesis organism like plants, cyanobacteria and algae. Plants, cyanobacteria and algae, they are the main stored food in plants. Okay, plants primary storage polysaccharide is the starch. Yes, primary storage. Okay, the main product of photosynthesis, what? Glucose. This glucose convert into starch for their storage process. That's why in their potatoes and some maize mean grains, they always stored as a starch. Clear? Very simple thing, right? Okay, primary storage polysaccharide is the starch. It is most important source of nutrition for humans. Yes, cereals, fruit, tubers, and some vegetable tubers mean this. But tubers, example for tubers. Cereals mean rice, maize type foods. Tubers, example for tubers. Yeah, sweet potato, right? Potato, sweet potato, yes. And some vegetables also, their main stored food is starch. Their main stored food is starch. It's a glucose polymer, yes. Alpha glucose, alpha glucose polymer composed of two components. This is the most another important point. Mainly the starch made with combining with two polymer types. Starch made by combining of two polymer types. They are amylose and amylopectin. Amylose and amylopectin. Okay, this is a two polysaccharide chains. Simply understand two polysaccharide chain. The combination of this amylose and amylopectin we called as starch. Amylopectin and amylose, the combination, the mixture we called as starch. Okay. It is alpha glucose polymer composed of two components, amylose and amylopectin. Remember, they are MCQ key points. That's a point to point I am explaining. Amylose is a water soluble component. Yes, amylose is simple monosaccharide chain. Amylose, okay, in the yeah, this is amylase. Amylase is a simple alpha glucose chain. Simple alpha glucose chain. Okay, chain alpha glucose chain. But amylopectin is a very complex structure like this. Amylopectin is very complex structure like this. See, this is amylopectin. It's a very complex structure, not a chain. It's complex and it's not water soluble. It's not water soluble. Okay, amylose is water soluble and it is 15 to 20% in starch. Okay, then remember the amylose percentage 20%, amylopectin 80%. Then that's why we call the starch is slightly water soluble. All polysaccharides are water insoluble, but starch slightly water soluble. That is slightly water soluble starch because 20% amylose. Amylose means this simple monosaccharide chain, alpha glucose chain. Okay, it is water soluble 20%. It's 20%. Then 80% is amylopectin. Amylopectin very complex, large structure. Therefore, it's not water soluble. But amylose water soluble. Therefore, only starch in polysaccharide slightly water soluble. Product clear? Clear? This amylose part is water soluble. Amylose is a long unbranched chain. Unbranched means it don't have branch. It's just only chain. Unbranched chain containing 200 to 1000 alpha D glucose. Okay, it can be vary to 200 to 1000 glucose. It's a very large chain. Now you can understand how about the difference between disaccharide and polysaccharide. See, only amylose structure, amylose chain, polymer chain, it's contain 200 to 1000 alpha glucoses. together by C1 to 4 glycosidic linkage. What's the meaning? Can you understand this point? It's contain it only amylose chain contain 200 to 1000 alpha glucoses with linkage with C1 to C1 mean carbon 1 to 4 glycosidic linkage. What's the meaning of this? Dehini. What's the meaning of this? Carbon 1 to 4 linkages mean? What's the meaning? Fatima? Carbon 1 to 4 linkages mean, can you understand this? This is the first carbon of this glucose. This is a fourth carbon of second glucose. Okay, that's why we called as carbon 1 to 4 glycosidic linkage. Carbon 1 to 4 glycosidic bonds. Clear? Okay, amylopectin is water insoluble in water, and accounts for approximately 80 to 85%. I told 80 to 85%. It can be vary. No, think it is 20 to 80. 20 amylose and 80 amylopectin. It's a branch chain polymer. Yes, it's a branch chain. It's a very complex. That's why it's not water soluble. Alpha D glucose unit with chain form 1 to 4 glycosidic linkage, and branches cost 1 to 6 glycosidic linkages. Okay, very important. Come to listen. Come to this part. Okay, they are the 1 to 4 glycosidic linkages. Glycosidic bonds 1 to 4, carbon 1 to 4 glycosidic bonds, as well as carbon 1 to 6. See, 1 to 6. These are branches made by 1 to 6 glycosidic linkages. Okay, the amylopectin, both 1 to 4 carbon 1 to 4 glycosidic bond, as well as carbon 1 to 6 glycosidic bonds. That's why it is very complex. It's a branch chain. Branch chain means like this. Simply understand, the starch is made by two main polymer chains. What are the names of them? Amylose and amylopectin. Amylose is very simple, like glucose chain, alpha glucose chain. It's just like this. It's only linear one. That's why it is water soluble. It's contain 200 to 1000 of monosaccharide alpha glucoses. Okay, it's only chain. But amylopectin is very complex. It's amylopectin not water soluble. It is this type chain, 1 to 4 chain, but it has branches as well. It has branches like this. Branches, it's a very complex. Very complex. Can you understand? Can you understand? Up to now, okay? Up to now, okay? Every point I explained, because in MCQ questions, they always ask. Kabir, up to now, okay? Any doubt? Any doubt? Dihini? Quickly. Today, at least we need to start our protein part. Amina? Okay. Okay, the starch is complete. Starch very simple, right? Starch is main. This is the function of starch is that's the main stored food in photosynthesis organism, and it's made by amylose and amylopectin two polymer chains. Amylose is a very simple one. It's the only chain. It's water soluble. It's only 20%. Amylopectin 80%, 80% amylopectin, and amylopectin is very complex, large, uh, linear polymer with branches. Linear polymer with branches. That means carbon 1 to 4 glycosidic linkage and carbon 1 to 6 glycosidic linkage both. Okay, this type simple knowledge you should have. Okay, cellulose. You know that the cellulose is the most available carbohydrate in the world. That's important. Most available. Why we call it is most available carbohydrate? Among carbohydrates, that's the most available because all plant cell, the cell wall main ingredient is the cellulose. All plant cells, the cell wall main ingredient is the cellulose. Cellulose is found in only in plant and is the most abundant. Abundant mean most available, right? Most available organic substance in the plant kingdom. It's a major component is a cell wall of plant cells. Yes, the main function, the main mainly available in the major component in plant cell wall. Cell wall. The plant cell wall main component is the cellulose. The main building blocks of the main building blocks of plant cell wall. Okay, the cellulose. Cellulose, you already know. Cellulose is a straight chain polysaccharide made up in entirely beta D-glucose. Beta D-glucose means beta glucose. It is made with beta glucose units linked together by a glycosidic linkage. Carbon 1 to 4. Yes, carbon 1 to 4, right? Carbon 1 to 4, but it is made by beta glucose. Remember, starch mainly made by alpha glucose. Amylose and amylopectin both made by alpha glucose, but cellulose mainly made by beta glucose 1 to 4 glycosidic bond. Beta glucose 1 to 4 glycosidic bond. Beta glucose 1 to 4 glycosidic bonds. Okay, that is the cellulose structure. It's like this. It's a chain, but it is not linear. It's not this type. It is like this because they are beta glucose. Beta glucose. They are beta glucose chain combined with glycosidic bond 1 to 4 glycosidic bonds. That's why it is. I think you can understand this glycosidic bond that we already discussed. Cellulose one. Do you remember cellulose? Cellulose එක අපි යන්දන්නේ ඒකෙම polymer chain. I think you can remember, right? Cellulose, beta glucose. Cellulose is made by beta glucose plus beta glucose. Remember? This one. This is a cellulose. Make the continuous version. The continuous version is the cellulose. Clear? Okay. What's the main function of cellulose? This is the main ingredient. The main compound is a plant cell wall, and this is a major abundant, mostly available carbohydrate types in the earth due to this plants availability of plants. Okay, that's all about cellulose. It is made by beta glucose 1 to 4 glycosidic linkage. Beta glucose 1 to 4 glycosidic linkage. Okay, if you have any doubt, please ask, because this lesson you should have wide knowledge for understand other chemistry lessons and biology lessons. Okay, any doubt, please ask. It's actually easy lesson, but you should have deep idea. Okay, the third one, glycogen. Third one, glycogen. Glycogen. Okay, I told, do you remember? I told main photosynthesis organism, mainly photosynthesis organism. Photosynthesis organism, the main stored food is starch. The main stored food is in photosynthesis process mainly produce glucose. In the storage process, this glucose convert into starch and store in the plant, tubers and different parts, roots, they store. They store. And if you got non-photosynthesis organism like us, non-photosynthesis organism like us and animals can't make photosynthesis, and uh, bacteria, heterotrophic bacteria mean they can't make foods. Only cyanobacteria are not heterotrophics, therefore they can make they are phototrophic. Okay, we called as cyanobacteria phototrophic bacteria, that we can discuss in next lesson, cell structure lesson. Bacteria, normal bacteria, they are heterotrophic bacteria. Heterotrophics. These heterotrophic bacteria cannot be make their own foods like us. They depend on other source for carbon and light. I will explain in next lesson. Okay, however, this all bacteria and all fungi and protozoa, animals and humans also type of animal. Okay, all are heterotrophics. This all heterotrophic organism, all non-photosynthesis organism, the main stored food is glycogen. But you need to remember, photosynthesis organism, the main stored food is starch. Non-photosynthesis organism like us, the main stored food is glycogen. The stored as glycogen animal body. Animal body mean non-photosynthesis. Okay, it's also known as the animal starch due to its structure, which is like amylopectin but more highly branches. Okay, the structure is like amylopectin. That means 1 to 4 glycosidic bond and 1 to 6 glycosidic bond both, but it is highly branched. Highly branched means that's very complex. Therefore, glycogen is not water soluble, completely not water soluble. It can be found in the liver, muscles, and brain. Yes, glycogen is in our body, liver, muscles, and brain. We can find this glycogen. When the body required glucose, enzymes can convert. That's the point. In our body, the main stored food is glycogen. When any requirement, they can store in muscles. In our muscles, මේ වගේ මේ තියෙන muscles වල store කරලා තියෙන්නේ as glycogen. Okay, the remain part, when we take foods, some required part is absorbed to the body, and the remain part is stored. Remain part එක store කරනවා. Then if any requirement, what happen, enzymes in our body hydrolysis, break down this glycogen into glucose. The glucose is our main energy source. I think you can remember, enzymes convert glycogen into glucose. Glycogen is found in yeast and fungi as well. Yes, glycogen, I told, yeast and fungi also, and bacteria also, heterotrophic bacteria also. The main stored food is the main stored food is glycogen. The main stored food is glycogen. Okay. The final one, inulin. The fourth polysaccharide, inulin. Inulin, the basic building block of fructose. I told, remember this glucose, galactose, fructose. Sorry, starch. Sorry, starch, cellulose, and glycogen. Their main building block is glucose, alpha or beta glucose. If you got starch, it is alpha glucose. If you got cellulose, it is beta glucose. Glycogen, alpha and beta both, but inulin, their main building block is fructose. Inulin main building block is fructose. Okay, it is given in the table, fructose. Inulin main building block is fructose. The basic building block is fructose. It is act as a reserve food in plants of the sunflower genus. Sunflower group, sunflower plants the main building block is this inulin. Dahlia tubers are also available as reserve foods. Dahlia tubers, it's only a few group of plants. The main stored food is, reserve food is inulin. Okay, that's not the important one. This starch, glycogen, and cellulose, very important polysaccharides. Okay. Any doubt up to now? Any doubt? Everything clear? Fatima? Okay. Okay. Main characteristic of polysaccharide. Now, at the initial part of the today class, we discussed main characteristic of monosaccharide and disaccharide. Okay. Now I am going to compare this monosaccharide and disaccharide with polysaccharide. Okay. Okay. The first characteristic, if you got monosaccharide and disaccharide, not polymers, both not polymers. No. If you got polysaccharide, they are polymers or not? Are they polymers or not? Are they polymers or not? Are they polymers or not? Yes, they are polymers because they are very large structures, polysaccharide කියන්නේ they are polymers, right? They are polymers. All 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 are polymers. Okay, how about the taste? Taste, is it sweet in taste or not? How about taste? Not sweet in taste. If it is sweet in taste, we can eat plant leaves, like we can eat, not sweet in taste, because cellulose not sweet in taste. Third one, are they crystallable? No, cellulose, glycogen, they are not crystallable. Not crystallable. Not crystallable. Not crystallable. Fourth one, if you got water solubility, are they water soluble? Are they water soluble? Not water soluble. Not water soluble. But within bracket you can mention only starch slightly water soluble, right? Because starch is made by amylose and amylopectin. Amylose 20%, 20% is water soluble. Then starch slightly water soluble. Starch slightly water soluble. Starch slightly water soluble. Okay, fifth one. Can we hydrolyze this or not? Before that, are they reducing sugar or non-reducing sugars? Yes, all are very complex sugars, right? All are non-reducing sugars. All are non-reducing sugars. Non-reducing sugars. All are non-reducing. All are non-reducing. Okay, final one. Can we hydrolyze or not? Fatima? Can be hydrolyzed or not? Can or can't? Hydrolyze means breakdown into simple compound. Yes, we can hydrolysis. Can be hydrolysis. Okay, in disaccharide, I told we can use different types of special enzymes as well as acid types for this hydrolysis process. Acid mean powerful acid, diluted versions. Diluted versions. Okay, now this is a very interesting story. Okay, write down. I think you have two rules, right? Example. Starch can be hydrolyzed by amylase enzyme. Amylase enzyme into alpha glucose. Alpha glucose. Amylase is the enzyme, not amylose. Amylose is a type of polysaccharide. Amylase enzyme. Amylase enzyme. If you got sucrose, no, cellulose. into beta glucose, we can hydrolyze by cellulase enzyme. Cellulase enzyme. Cellulase enzyme. Cellulase enzyme. Cellulase enzyme. Cellulase enzyme. It is into beta glucose. The building block is again converted into building blocks. Okay, do you know we don't have feel to eat grass, like grass, plant leaves, we can't eat. අපිට feeling එකක් නැහැ. But cow, goat, elephant, they can eat. What's the reason? ඇයි අපිට eat කරන්න බැරුව එයාලට eat කරන්න පුළුවන්? Yes. In our digestive system, there is no any cellulase in our digestive system. අපේ තිබ්බනම් okay, but in our digestive system don't have any organelles to secrete cellulase. But in our digestive system අපිට මොනවද තියෙන්නේ? Amylase. The amylase is the main enzyme involved for our digestive system. But in elephant and cow, this type animals, what is the main enzyme? Main enzyme is the cellulase enzyme. Cellulase enzyme. That's the main reason. That's the main reason. We normally in our digestive system we can digest very smooth high cellulose parts. Smooth cellulose අපි ඔය green leaves කියලා අපි පලා ජාතිකන්, they are very smooth parts, very immature parts. They are not complex cellulose. But these animals can digest complex cellulose, but we can't. In our digestive system only secrete amylase enzyme. Amylase විතරයි. That's the main reason. Okay, that's the reason. However, the nutrition type same. Okay, then අපි rice කෑවත්, elephant cellulose කෑවත්, the nutrition type same because same glucose. Finally, it is converted to glucose, but the breakdown type is a different type. Okay, any doubt up to now? Okay. Any doubt up to now? Everything okay? Okay, everything okay? Oh, sorry. Hari. Okay, that's a significance that they are the main functions of carbohydrate actually. Simple. It's a energy source, you know, the main energy source. Give me a second. Okay, sorry. Yeah, significance as energy source, you know, in the main energy source in our body and microorganisms, the main type is glucose, the main sucrose, and obtain energy for activities for of organism. It's a mistake. Organism is the carbohydrate, the monosaccharide glucose main. Glucose produced due to the hydrolysis of these compounds release energy. The main energy source is glucose. Therefore, carbohydrate is the main energy source is organism and all animals and microorganisms. As a storage compound that you already know, the starch in photosynthesis organism, the main storage compound is the starch, main stored food. In non-photosynthetic organism like us, the main stored food is glycogen. Okay, therefore it is a storage compound. As a structural compound, cellulose, mainly in plant cell cellulose, it's a structural compound. As a of nucleic acid, just for hereditary, for transformation of hereditary materials. Hereditary materials, the main because ribose, tribulose, deoxyribose, ribose, tribulose, deoxyribose, they make DNA and RNA. DNA and RNA. DNA and RNA, they this monosaccharide important for this hereditary materials, DNA and RNA. Okay, that's a carbohydrate lesson. No doubt, right? Fatima, Kabir, everything clear up to now? Okay. Amina, Joel? Okay. Okay. Okay, this is another very important part you need to understand that we called as carbohydrate identification test. Carbohydrate identification test. In this test, our main target identification of reducing and non-reducing sugars. Reducing and non-reducing sugars. Now, I think you have idea what are the reducing sugars and what are the non-reducing sugars. Okay, sugar, we divide into two types, reducing sugars, non-reducing sugars. What are the reducing sugars? All monosaccharide, right? All monosaccharide, glucose, galactose, fructose, ribose, ribulose, deoxyribose, all monosaccharide, all monosaccharide, all monosaccharide, and all disaccharide except sucrose. All disaccharide except sucrose. I think you can remember. Then with sucrose plus all polysaccharide, all polysaccharides, they are non-reducing sugars. Non-reducing mean they are very complex sugars. Okay, they are the reducing and non-reducing sugars. Our main target of this carbohydrate identification test to identify this reducing and non-reducing sugar. Okay, that's very important part, important practical. It's bit advanced. Try to get your maximum idea. Okay, first this test we called as Benedict-Felix test. Benedict-Felix test. Benedict-Felix test or carbohydrate identification test or reducing sugar identification test or non-reducing sugar identification test. All names are correct. Okay. Before we move into this reducing sugar test, this is very simple test that you already know. That's starch identification test. We can separately identify starch by using iodine that you already know. I think in O-level time, that's called as starch test. Starch identification test. Small amount of food is obtained from and grind with well water and drop of iodine. Normally, what is the color of iodine? What is iodine color? What's the color of iodine? What's the iodine color? Yes. It's in brown color, right? Very good. Wait, I will show you these practicals. Then it is simple. Okay, give me a second. I will show you this practical. Give me 1 minute. Oh, okay. They now. Okay. I'll show these videos because they are in my another lab, like my other laptop. That's why I couldn't find this. Okay, I'll get it. Okay, however, now I will explain. After that, we can see these videos. Okay, the starch identification test, you already know. If we need to add few iodine drops. Initial, the color of iodine, it is brown. Then if it is contain starch, what happened is it is converted to blue color. It's converted to blue color. Okay. Okay, I will find these videos. If you want, you can get a break. 10-minute break. Again, we can start now 8:34. We again start 8:45. Take a break and come again. I'll find these videos and we can discuss these practicals and we start again. Next we start proteins. Okay, take a break and join 45. Okay. We discussing about carbohydrate identification test. The first one is the starch identification test that we already discussed. I told starch identification test we use iodine. Iodine is a reagent we can use for this starch identification test. Then initial color of iodine, it is brown, yellow to brown color. It's converted into blue to blue black color. See,
simple carbohydrates are the sugars found in anything sweet. Starchy carbohydrates are found in foods like pasta, potatoes, cereals, and rice. A few drops of iodine solution
this is the iodine.
color change from orange to blue.
This is the initial color of iodine. It's brown.
Black. This indicates the presence of starch. So, will any of our foods test positive? Grape juice? Nuts? Or bread?
The bread. What
starch is present in bread, but not in grape juice or nuts.
Okay, this is the test. If it is present starch, what happened? It's converted to purple to blue. Actually, it's very dark color. It's like black actually. Blue black color converted into blue black color. Okay, clear? Iodine test. Are you clear the iodine test? Dihini? Okay, next move into our reducing sugar identification test. This is the most important one. Reducing sugar identification test. Reducing sugar identification test. This is a method. Try to get the full idea. Okay, first step. This is I'll explain it step by step. Then we can watch with videos for get practical idea. Okay, the first thing I need to confirm. Think I have a food sample. Okay, food sample like bread or something. I have food sample. I want to confirm. I want to check whether it food sample include reducing sugars. Reducing sugar mean you already know all monosaccharide and except sucrose, all disaccharide. Okay, this is the identification test. The test we called as Benedict or Felix test. Benedict or Felix test. Okay, what we need to do, first of all, you need to prepare a water solution of this sample, food sample, convert into dissolve in water. Dissolve in water. Food sample dissolve in water and convert into water solution. And the second step, add into this type test tube. Like this. Test tube. Next, second step done. Third step, we need to add Benedict or Felix. Both regions we can use Benedict or Felix. Benedict or Felix. Benedict or Felix. Few drops. This is a solution. This is a blue color solution, light blue color solution. We need to add few drops, 2 to 3 drops. 2 to 3 drops of Benedict or Felix. දෙකම ඕනේ නැහැ. Benedict or Felix. Okay. Benedict or Felix. Few drops. Few drops. Benedict or Felix. Okay, third step done. What is the first step? Put sample, make as a water solution. After that, after that, add into a test tube and add Benedict or Felix 2 to 3 drops. Next one, heat the container by using water bath. You can't directly heat. You have to use a water bath. Water bath. This is a use Bunsen burner, and we need to heat this water bath. Then what happened? What happened? This initially it is blue in color because of this Benedict or Felix color. It is in blue in color. Blue in color. Initially it's blue in color, but if it is contain reducing sugar monosaccharide or except sucrose disaccharide, if it contain reducing sugar, what happened? This blue color, when we gradually heating, we use water bath for uniform heating. Uniform heating, I will explain later. Okay, if it is contain reducing sugar, what happened? This blue color convert into green, yes, green. Then after few seconds, it's convert into yellow. It's like a magic yellow. Then after few seconds, it's converted to orange. Finally, orange එක convert වෙනවා brick red color. Brick red color. Brick red color. Okay. Let's see this practical. Okay, this is the test.
We are going to perform the Benedict test. The apparatus used for this test are test tube stand, a test tube holder, measuring cylinder, and a beaker containing the sample. The reagent used for this test is Benedict's reagent. Take a test tube and hold it firmly with the test tube holder. Add 5 ml of Benedict's reagent.
This is a Benedict color, light green, sorry, light blue color, blue in color.
Add 8 drops of the sample.
This is a sample. That can be changed with our procedure. Our procedure, we need to add a few drops of Benedict's into the sample. This opposite direction, no problem.
Shake the contents well and boil it for approximately 1 minute.
Okay, this boiling method wrong. Okay, according to our syllabus, we need to boil with water bath. I will show you. See the color variation.
Shake the test tube by heating it.
Okay, now you can see this test tube, blue color. Now you can see it's converting to green, right? Green. Now it's green. Green.
There is a formation of a color change, which indicates the positive Benedict test.
Now it's don't have color change. It's green color. Wait, I will show you the color variation.
GYOR, green, yellow. So orange, red, and brick red precipitate. See the changing color. Now it is green.
Green. Okay.
Now it is moving towards yellow, then orange, then red, and brick red.
This is bit orange. So red, yellow. Now it is orange. Okay. This is a color variation.
Orange.
This is glucose. This is a Benedict color.
Followed by adding and people.
This is our sample. Food sample. This is a glucose solution. Yeah, this is the correct way. This is at 3 Benedict drops. Okay, then yeah, this is the correct way. Take pill and heat by a water bath. This is a water bath. Water bath, Bunsen burner, beaker. Yeah, this is a correct way. Observe the color difference.
Observe the color change. Original color now is
Light blue is the original color. Okay. What happened? Gradually convert into green now. Green convert into yellow. Yellow. Now it's bit orange. Finally, it's make a brick red. Brick red color is precipitate. Brick red mean very dark red color. See this one, very dark red color.
Once
This is
it
the
turned
final
into
color.
red, the and now you can see the orange tree.
Okay. The final one is our all the steps correct. Okay. What we need to do first, make a solution of the food sample, then add few drops of Benedict solution, Benedict or Felix we can use, then heating by water bath. Then if it is showing normally light blue color, convert into green, yellow, orange and brick red precipitate. Brick red is a precipitate. Brick red precipitate we call this is a it contain reducing sugar. This food sample contain reducing sugar. If not, we can say this food sample not contain reducing sugar. Okay, understood and clear all the steps. Are you clear all steps? Abir? Okay, Dihini, all are clear? Okay. Okay. Test for reducing sugar. A solution of glucose is obtained into test tube. Glucose or whatever food sample. Okay. Few drops of Benedict solution add to the above solution is added. Okay. The above solution is immersed in a water bath and heated. This water bath is very important. Why we need to get accurate observations? Why we need to use water bath? Why we need to use? What's the requirement? What do you think? Okay, without we heating by direct Bunsen burner, why we need to use water bath for heating process? In final video, this one, why we need to use this type water bath? Yes, evenly spread the heat. Yes, uniform distribution of heat. Okay, in previous videos, they are using just simple method. Then it's directly heating, no? Directly heating, what happens? Some part of the test tube is overheating and above part is not heating well. But if you use water bath, then we can uniformly heat the test tube or the reactant. That's why heating by water bath. Okay, do you know why we heating actually? What's the purpose of this heating? ඇයි අපි heat කරන්නේ for this reaction? What's the purpose of heating? In previous kinetic chemistry lesson, you can understand why we need to heat. What's the requirement? ඇයි අපි මේක heat කරන්නේ? No break bonds. Bond break වෙන්න දෙයක් නැහැනේ. Joel, we testing non-reducing sugars. Non-reducing sugars mean glucose like simple compounds. They no need to break bonds, right? Why we need to heat? What's the purpose of heating? What's the purpose of heating? Amina? Amina? Kabir, why we need to heat? Fatima? What is the requirement? Actually, if this Benedict is a reagent, no, that is reacting with this food sample, reducing sugar, this color change is made. Color change එක හදනවා if we not heated. But why we need to heat this? We heating for increase this reaction rate. When we reacting this color change, we can observe this color change. Are you good? Do you remember in kinetic chemistry lesson? The temperature is a factor, main factor, direct factor effect on rate of chemical reaction. While we heating, what happened? The rate of chemical reaction increasing. That's why we can observe this color change within few seconds. If not, the reaction take place, it take long time. Understood, Fatima? Heating to increase the rate of chemical reaction. Now we already know. In kinetic chemistry, we discussed. Okay. Can observe color change below. Blue converted to green, green yellow, orange, and brick red precipitation. Brick red precipitation is our final observation. If this observation we can observe, if it is showing this observation, then we can decide the food sample contain reducing sugar. Okay, test for non-reducing sugar identification test. Non-reducing sugar identification test, the most important part of the lesson, the most important practical. Okay, understand what I told previous. Non-reducing sugar identification test. Okay, the first step, food sample make as solution එකක් විදියට හද. Second step, add to a beaker and add this solution. Add few Benedict drops. Same as previous. Add few Benedict drops. Okay. We go test tube. Okay. No, it's test tube. It should be test tube. Test tube and add few Benedict drops. Okay. Third step. After we add this Benedict solution, Benedict drops, them mama, after we add these Benedict drops, the third step same as previous. Third step, heating by water bath. Heating by water bath. Okay, heating by water bath. First, what we're doing, it is same as previous. Make as a solution. Put sample, make as a solution. Then add few Benedict drops and heat by water bath. Okay, when we heating, if it contain reducing sugar, if it's contain reducing sugar, if it's include contain reducing sugar, what happened? We can observe the color variation and precipitation, right? We can observe the precipitation. If don't have no precipitation, no color variation. Okay, reducing sugar skip both, we can observe color variation. Now what we going to do, if it is showing contain reducing sugar, color change එකක් වෙනවනේ. It's show the color variation. Show the color variation and make a brick red precipitation. Make a brick red precipitation is made. Then what is my next step? I need to remove this. I need to filter out this brick red precipitation. Brick red එකක් ආවොත් අපි මොකද කරන්න ඕනේ? We need to filter out this. Filter out this brick red precipitation. Precipitate එකයි අයින් කරන්න. Filter out this precipitation. Filter out this precipitation. Are you clear up to now? What is my first step? Simply, what I need to do, conduct the reducing sugar identification test. If it is show any color variation and precipitation, what I need to do, filter and remove. We can use filter papers. Okay, filter papers, and we can remove this red precipitation. Okay, that is the first step. This is the first step. Okay, after that we can start our non-reducing identification test. Okay, white color part is the reducing sugar identification test. This is a fifth step. Up then filter out karla tiyena. We have already filtered out this precipitation. Therefore, that don't have reducing sugar now. Okay, that don't have reducing sugar. Reducing sugar filter out reducing sugar. Okay, our fifth steps. Add few diluted HCl drops. Most important. Now you already know what is our target. What's our target? To identify non-reducing sugar. For identification of non-reducing sugar, actually we don't have non-reducing sugar identification test. Chemical use color we can't identify by a chemical. Therefore, I'm going to convert. Now I already filtered out this reducing sugar. Now I'm going to convert this. The non-reducing sugar contained is in this food sample. I'm going to convert into reducing sugar by hydrolyzing. I'm going to convert this non-reducing sugar into reducing sugar. Okay, now I'm going to add few diluted HCl drops. Six steps. Add few diluted HCl drops. Sixth step. Heat by water bath. Heating by water bath. Okay, what is the purpose of this heating? This is step heating. මේ වෙලාවේ heat කරන එක function එක. What's the purpose? Remember the heating purpose of this heating to increase the breakdown speed, to increase the hydrolysis speed. I told we can hydrolyze any polysaccharide, any disaccharide or sucrose or any polysaccharide we can hydrolyze by acid. Okay, this acid doing their process, doing their job, but we can increase their function, increase the rate, this hydrolysis rate by using heating. The heating means increase the chemical rate, increase the rate of chemical equation. Okay, I'm going to use this purpose, heating by water bath. Heat karanna ya. Okay, after heating, 7 step. After heating, now already breakdown. Okay, already breakdown. After breakdown, 7th step, what I'm going to do, I need to add few drop of sodium hydroxide like base. Sodium hydroxide, it's a base. Okay, what's the purpose of sodium hydroxide? Remember, these Benedict's and Felix regions only reacting, only doing their job, only doing color variation in neutral conditions. Neutral condition, pH 6.5 to 7.5 like neutral conditions. But when we add HCl, what happened? The pH value getting very low. The acidity of the solution increasing. Acidity වැඩි වෙනවා. That's why we need to add sodium hydroxide for neutralize the solution. Neutralize the solution. Okay, I'm going to my final steps. 8. Need to add heat by water bath. Sorry. Again add few Benedict drops and few Benedict drops and heat by water bath. Heat by water bath. Okay, this is the full practical of non-reducing sugar identification test. Okay, tell me with, okay, revise again with me. What is the first thing? First, you need to test the reducing sugar identification test. If it is showing color variation, precipitation, what you need to do? Any doubt? Right. Oh, sorry. Okay. Okay, first check the Benedict test, reducing sugar identification test. If it is showing color variation with precipitate, I need to filter out this precipitation. Then we can remove all reducing sugars. Now I'm going to our non-reducing sugar identification test. First, what I need to do, this non-reducing sugar identification, we don't have any chemical. Therefore, what I need to do, this non-reducing sugar by hydrolysis, I convert into reducing sugar. Yeah, breakdown on think starch or cellulose. If you're going to identify cellulose, what happened? Breakdown into their initial compounds, building blocks. They convert into beta glucose. Beta glucose reducing or non-reducing? Reducing. They are simple. Okay. Then add few diluted HCl and heat it. This is hydrolysis process. Hydrolysis process. Okay. Third step, we need to add few sodium hydroxide solution for neutralize. Finally, again add Benedict's drops and heating by water bath. In these final steps, In final step, if it is again show this previous color variation, finally again convert into blue, light blue convert into green, orange, sorry, green, yellow, orange, and brick red precipitation, we can decide this sample contain non-reducing sugar. This food sample. Any doubt? Or do I need to explain again? It's very important part. Any doubt? Dihini? Okay, you have to remember this. Okay, try to understand by video.
So when we're testing for non-reducing sugar, you always need to make sure that you carry out the
Okay.
So when we're testing for non-reducing sugar, you always need to make sure that you carry out the reducing sugar test first, so that you can eliminate whether or not the sample is actually a reducing sugar. You wouldn't know if you did the non-reducing sugar test first. So this is the result of testing two samples, A and B, with the reducing sugar test, and we saw that sample B gave us a negative result.
This is the result of reducing sugar test. The test is doing from 4th steps. Okay. This is the result. Then this sample don't have reducing sugar. This sample contain reducing sugar. Understood?
But it remains blue, indicating to us that sample B is not a reducing sugar. So if we take sample A away, we know that that's a reducing sugar because it turned orange. So we don't need to go any further with that sample. So sample B, then we need to continue to find out whether if it's actually a non-reducing sugar instead. So really important that we discard this sample and we now take a new sample of our solution B. So a fresh sample of B in our tube. And the first thing we need to do this time is add some.
Okay. Now think this is the sample after filter out. Oh, think it don't have any color variation in the first step. Okay, this don't have reducing sugar. Now I'm going to check non-reducing sugar. Okay.
Hydrochloric acid.
This is hydrochloric
We're
acid.
going to heat the
HCl.
sample up again after adding our hydrochloric acid. Okay, so hydrochloric acid added to our solution. and we're placing it back into the water bath. And we place it back into the hot water bath for around about 2 to 3 minutes. So it's really important that this water is as hot as it can be, and so the acid starts to work. And we'll talk about what's happened and what that acid is actually doing to the sample in a sec. Okay, so 2 minutes has passed, and what we need to do now is remove our doing to the sample, and we need to in a sec. Okay, so 2 or 3 minutes has passed, and what we need to do now
what's the purpose of this heating? To break down bond and hydrolysis. That's the purpose. To break down bond and hydrolysis. Okay, then
to remove our sample, and we need to neutralize that acid. So we
neutralize
the acid with an alkaline, and we tend to use sodium
hydroxide.
Carbonate.
Sodium hydrocarbonate, or whatever base you can use, they use sodium hydrocarbonate. If not, you can use caustic soda, sodium hydrocarbonate.
For your exam, it really doesn't matter if you can't remember that name. What's important is that you remember it's an alkaline, and its purpose is to neutralize the hydrochloric acid. So we just add half a spatula full in there, and as you can see, as we add the alkaline, it starts to fizz up a bit. So the idea is that you add the alkaline until the solution doesn't fizz anymore, and you know that that has been neutralized. And then we finally add our Benedict's solution again. So if you remember, the Benedict solution was a blue color. So we add Benedict again to our solution before reheating it. And again, remember, it's important to talk about the temperature. We heat it to above 60 degrees. So originally we added the acid, and you have to boil the sample with acid. So boil with that.
Okay, Ms. Abraham.
Acid, you heat with Benedicts. So we'll just leave that sample to develop for 2 to 3 minutes again with the Benedicts in our hot water bath. Okay, so about 10 minutes has passed.
What happened? It's only non-reducing sugar sample convert into brown.
And we can see that after that time, our sample B has now changed to that orange, nearly brick red colour. So this was our sample B after the reducing sugar test. It stayed blue, indicating that sample B was not a reducing sugar, but we took a fresh sample and we added a hydrochloric acid, and we boiled with hydrochloric acid. We then neutralised the hydrochloric acid using sodium hydrocarbonate, which is an alkaline. And then we added our Benedict solution again, and we placed our sample back into our hot water bath, and we heated the sample with the Benedicts. And on the second occasion, you can see that we have a positive result. The solutions turn from blue to orange. So this indicates that sample B was in fact a non-reducing sugar. And the non-reducing sugar that you need to remember for your exam is sucrose. So sample B must be sucrose. There are no other non-reducing sugars that we see. Now, this is a little bit of an extension thinking about why we actually see this result. So initially, we had a negative result because there was no reducing sugar present. But if you think about what sucrose actually is, sucrose is a dye.
I got this. Any doubt of this video? මේක තේරුණාද මෙන්න මේ test tube දෙක වලට පැටලෙන්නේ නැති මේක this is a reducing sugar test. Reducing sugar test, that means this food sample,
The sucrose is actually made up of the two.
this food sample don't have reducing sugar, but after we add few Benedict drops, few HCl drops and break down, again neutralized by sodium hydroxide or sodium bicarbonate, what happened? If you use Benedict test again, it's showing color variation. What's the meaning? This food sample don't have reducing sugar. Glucose, like simple sugar, don't have, but they contain non-reducing sugar, like cellulose, glycogen, whatever they contain. Understood? Any doubt? No, we can discuss, because today we're not going to start protein. I hope to complete everything about carbohydrate, every parts. Any doubt? Fatima? Others, any? Amina? Are you clear? Step by step, you have to remember. All parts, step by steps, need to be remembered. See, go to tube. Sucrose solution is obtained into a sucrose. Then අපි ගන්නේ sucrose. Sucrose reducing or non-reducing? Non-reducing. Okay. Sucrose solution is obtained into a test tube. Few drops of Benedict solution added to it. The test tube is immersed in water bath and heated. No color change. Why don't have color change? Sucrose is not a reducing sugar. අපි sucrose කියලා sugar solution එකක් ගත්තා නම් හරි. That's sucrose. Okay, you can. Okay, let's try to do it in practically. Practically, the same thing. Okay, yeah, iodine we have, and Benedict. If you can Benedict solution, I will try to do it next week. Do it in practical. But the same thing, you can understand. Few drops of diluted sulfuric acid, sulfuric or HCl you can use, no problem. Whatever acid added to freshly prepared sugar solution and heated. Heat karane, what's the purpose? What's the purpose? Break down bond and hydrolysis. Okay, it's very simple. After hydrolysis, what happens? Non-reducing sugar, complex sugars convert into simple sugars, convert into monosaccharides. All monosaccharides are reducing sugar. Then monosaccharide, what happens? Add few drops of Benedict solution. It's one step is missing, right? Yeah. Please write down one step missing. Add few drop of sodium hydroxide base for neutralize the solution. For neutralize the solution. Okay, please note down. Point after heating, we need to add few sodium hydroxide drops. After that, add new few Benedict drops and observe color change below, same as previous. Okay, then we can conclude, we can decide the sample, the sucrose sample contain non-reducing sugar. Okay, any doubt? Any Habib? Any doubt? Any doubt? Okay. Add few drops of sodium hydroxide base. So sodium bicarbonate you can use. Whatever base no, for hydrolyze, for neutralize the solution. After that, add Benedict and heat it. If it's showing again this briquette precipitation, we can say the food sample contain non-reducing sugar. Okay, the carbohydrate is completed. If any doubt about carbohydrate, you can ask now. Okay, actually the 50% of this lesson carbohydrate. The proteins, lipids, easy. Dihini, everything clear? Okay. Okay, actually we did last week part. Okay, this other today part we can discuss. Next Sunday we doing paper as well, our refresh paper as well. Now I think you can refresh number 1 and 2 we already discussed. Refresh number 1 and 2. Both papers have some I don't have refresh number 2. No biomolecule questions. Okay, I will yeah, after this we doing, I hope to do rapidly first lesson. After that we doing work energy lesson. After that mechanical properties of matters. I will I'll make the schedule and share with you what are the lessons we revise in the revision class, and we're not going to discuss lessons after coordinate geometry we did in theory class. We're not going to revise again because coordinate geometry, those lessons we're not going to discuss again. But measurements, I think measurements also we don't need to discuss again. Measurement is in first lesson. I hope to discuss all the important parts only. Rapidly we doing, not like this. This is a full theory lesson, and this lesson we did as our third lesson, as I remember, in 2026 class. That's why I'm doing in this everything again. But fourth lesson we're not going to do like this. We're doing rapidly and we're doing a lot of questions from first lesson. And this lesson also I have 2015 to 2024 separated all questions I have separated all question in biomolecule lesson. If we discuss this paper after this lesson, everything will be covered. Okay. Okay, then let's meet again Sunday, 2 p.m. with our new revision. Actually, the missing part I discussed, carbohydrate part, our previous week part. Again, we start the protein in next Sunday. Next Sunday means day after tomorrow. Okay, any doubt from today part? No, right? Okay. Goodbye, good night. Let's meet Sunday.